Thank you, Suraj. Thank you, Ganga, for putting this. Am I audible? Okay, great. So this is something that I put together fast. I mean, I teach a whole course on pattern formation, so I just pick things here and there. If there's some lack of coherence, you're welcome to come and bug me anytime. So have people studied patterns? Just show me a raise of hands. Okay, not many. Okay, great. So I just want to get a couple of pictures and so that we're all on the same slide. So is this a pattern? Yes, no, maybe, I don't know, I don't care, something. Yes, okay. Is that a pattern? Is that a pattern? Is that a pattern? Is that definitely a pattern? Okay. What about that? Yes, no? 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 Maybe, okay. Okay, okay, so let's go back. That's a pattern. Sure. All right. What about that? That's a pattern? Yes, how many? Okay, and the others know how many? The others, I don't care. I mean, it's a hard thing to define. I mean, so there's no definite answer. So, I mean, there's some regularity, surely. It should be non-random, something which is totally random, you probably won't call a pattern. But you can have small asymmetry. I mean, this, most of you did call it a pattern, right? Because you could see remnants of a nice periodic crystalline lattice, but there were some small distortions coming up. Okay, I want to connect that to some other idea and think about symmetry. Again, people have heard of symmetry, thought about. So I want to show a boring graph. So f of x is a constant. Okay? Absolutely boring. So I ask you the following, that if I look at some position here and I move freely on the x-axis, does the plot look different? It looks the same everywhere. What about this plot? So it looks like all the points on the x-axis are the same. And the function overlaps with itself, no matter where you are on the x-axis. Any infinitesimal displacement. Is that right? So this continuous translational symmetry, continuous. Any infinitesimal dx, the plot overlaps with it. What about this one? So does it overlap with itself for any discrete, any infinitesimal dx displacement? No, right? So all points are not the same. And so the function overlaps with itself only when you do discrete displacements like 2n pi, right? So there's discrete translational symmetry. So I want to put it in a different language that you're breaking continuous translational symmetry when you're going from here to here. Does that make sense? So typically, patterns break symmetry. I want to ask you this question. Of these two figures, which is symmetrical, which is a pattern? Now you need to get a sense of what is symmetry. Which one is symmetrical? Both are symmetrical. Is one more symmetrical than the other? How? Why? Okay. So I'm going to do a couple of rotations by some angles. So I did a rotation by 45 degree and well, okay, the circle overlapped with itself, but not the star. I'll go back, and if I do a 60-degree rotation, surely both will overlap with itself. Now it's easy for you to see the remaining things, right? The circle will overlap with itself for any arbitrary infinitesimal displacement, but not so with the star. So one way to put this is that the circle is more symmetrical than the star. In other words, the star results by breaking the continuous rotational symmetry of the circle. So what you saw in the previous slide was translational symmetry. This is rotations. Does it make sense? So one caveat that you can take is that patterns typically come because they break symmetry. Not always, but always. So there's no hard and well-defined way. You know it when you see it. And so some guidelines that you can pick up from some books. I mean, there's some form with some regular features that are recognizably and so on. But what are the kinds of patterns you see in biological systems, living systems? Okay. Surely you've seen all kinds of vegetable patterns or natural scale patterns and things. Must have seen even patterns on snakes. They're nice. There might be evolutionary reasons for that, but there are patterns. There are patterns which are, different on the up and down or along the length, stripes, longer stripes, parallel to the long axis. Apparently, there are snakes with all kinds of patterns that you can have. Animal coat patterns, classic examples that we all learn in school and college. Fish skin patterns. They're all examples of where there is some function, maybe a pigmentation, maybe a scale, maybe a shape, which is non-uniform in space. Not just space, there could also be patterns in time. For instance, many cells show rhythmic patterns in chemical concentration coupled to the cell division cycle or circadian rhythms on longer time scales. So though it might not look very symmetric, but if you follow a bunch of cells and you follow the time dependence of certain chemicals, you see a very nice periodic regular signaling that's happening. But then patterns need not be at a very gross, very big level. I mean, not just at the organismal level. Patterns appear even at the cellular level and tissue level. And I want to build a couple of ideas to think about patterns at the cellular and tissue level. Okay, so cell division is a classic pattern. For example, there's the spindle that forms, which is basically the microtubules which form in the shape of a nice two asters. They are the ones that are actually responsible for segregating the chromosomes once the chromosomes have duplicated. But there's also patterns at a much larger scale. For example, this is asymmetric cell division. Typical cell division that you learn in school textbooks is where the two daughter cells are the same. This is an example where the two daughter cells are not the same. They're not the same in multiple ways. If you notice for a minute carefully, you'll see the two daughter cells are different in size. This one is bigger compared to that. Secondly, there's also segregation of certain cytoplasmic components. So some molecule pi1e, which has been labeled with some blue color, and you see preferentially that goes to the right daughter cell compared to the left daughter. So initially I had something where I couldn't distinguish the head or tail. This and that axis was the same. But at the end of the patterning process, I have a way where I can say this direction is different from that direction. In other words, I can write an arrow. Does it make sense? Creatures, including us, use patterns like this to build your body axis. Head, toe, front, back, left, right. Every creature does this, including us. You see patterns at various scales. Tissue patterns, for instance. This is in the Drosophila fly, common fly. If you keep a banana peel open, you'll see tiny black insects. That's Drosophila. So that's a very nice model organism in developmental biology. People have studied loads and loads of things about its genetics from very early developmental stages. Recent work has focused more on large-scale patterns that come up. Can you recognize this structure? There's the wing. So you see this large-scale movement of cells. So this is, I don't know, I think several hundreds of microns, close to a millimeter or something. And you see something where there was a more or less homogeneous pattern of cells. As time progresses, this is in hours, you see a large-scale flow of these cells. And can you recognize some vein patterns in the wings developing? Right? Okay, so these patterns seem to come up spontaneously, of course, driven by certain signals. These spatio-temporal regularities are what gives you forms and shapes in the wing system. You can get patterns even at equilibrium. One thing I'll try to distinguish is the typical patterns that we'll study will be all driven out of equilibrium. I assume everybody knows what is thermodynamic equilibrium, thermal, chemical, mechanical equilibrium. Typically, at equilibrium, you don't get spatial dependence, except when you have phase transitions and so on. Here's another classic example. If you have amphiphilic molecules, lipids, for instance, so these are molecules that have a hydrophilic head and a hydrophobic tail. So the hydrophobic tail does not like to be around water. And if you put such molecules in water, they spontaneously form very nice patterns. There are liposomes, micelles, bilayer sheets, and so on. Here's a very nice simulation that you can actually do. You can do that right in your machines. So you don't need to put in the detailed chemistry of the lipid, water interaction, and so on. So let's say you just have a dimer, and you put a harmonic oscillator spring between them. And you just say water is just some spherical molecule. You make one of the heads of the dimer repel less by water and the other one repel more by water. That's it. And just put them in a box, run Newton's laws, and as I run the movie, you'll see a spontaneous emergence of micelles, liposomes, and bilayer sheets coming up. This is pure thermodynamic equilibrium, and these are the structures that actually minimize the free energy. Okay, you see a lipid bilayer coming up here, some vesicle-like thing coming up there, some micelles coming up. If you wait long enough, this will all fuse and give you one big structure. My patience ran out, so I didn't go that long. By the way, feel free to interrupt and ask questions anytime. So just to emphasize, thermodynamic equilibrium is when you have all three things satisfied. And there is, at least in principle, a well-established theory that we can do for systems which are thermodynamic equilibrium. The complexity increases as you have more and more complicated interactions. But nevertheless, there is in principle at least a theory. For a driven non-equilibrium system, we don't have a general theory so far. What we have is tools and techniques and tricks for which work for a particular class of systems. Unless, of course, you can write down the full dynamics and follow everything in detail, which we can't. Okay, so just to distinguish, pattern formation typically at equilibrium is something where you optimize the free energy. There's some scalar, so maybe some fields, some density fields, order parameter fields. that some combination of that gives you a scalar, the free energy. You optimize that free energy. It's reasonably easy. If you're far from equilibrium, there's nothing to optimize, right? So by definition, this is a system which is not isolated. There's a flux of energy. And so therefore, there's no free energy optimization. Typically, this is very hard. And you typically have to work with the equations of motion directly rather than trying to minimize the scale. But there are patterns far from equilibrium. Classic examples being Rayleigh-Bénard convection, which you should see in a kitchen. If you put it on a stove, beyond a certain temperature difference, conduction will become unstable and you'll start seeing convection rolls coming up. They can organize at much larger scale into nice hexagonal patterns and so on. Rayleigh and Bénard were one of the first who actually saw it. If you haven't seen this, can you guess what this is? If you have seen it, don't give up. Can you guess what this is? I've told you what it is, one Karman vortex feed. But what is the system you're looking at? Any guess? Huh? Smoke, yeah, one, anything? Cloud, anything? Ice sheets, okay. So this is actually a photograph from the Pacific Ocean. So it turns out there is a small island here, and this is several thousands of kilometers. And you see patterns, even von Karman vortex streets on thousands of kilometers. Does it make sense? Obviously, this is a driven system. There's heat, there's fluid. But you see patterns at very different length scales. So I showed you patterns at the cellular level. I want to show you some patterns coming up at the developmental level, how organisms develop. So typically, most of us in this room, maybe some bacteria apart from, are multicellular creatures. We typically start out by a single cell, which gets fertilized. And that's called the zygote. And once that zygote is fertilized, it starts dividing. 1 becomes 2, becomes 4, 8, 16, so on and so forth. And it just doesn't keep dividing. There's a whole mass of cells. But after some point, it starts rearranging itself in space and time. That's what gives you a typical form for a creature. I'll play the movie and we'll come back and discuss. So this is a zebrafish zygote, so about 0.5 millimeter sized object. That's the zygote, a single cell which has been fertilized. That's the yolk, which is the food for the creature. So that's the energy supply for the creature until it becomes big enough, can swim and fend for itself. As you see the movie, you see cells dividing. A ball of cells has formed, starts spreading. At some point of time, it starts elongating. And about 20 hours down the line, you see the appearance of a fish-like structure coming up. Just like us, this is a vertebrate. So you see a periodic segmentation patterns coming up there, just like the vertebrae that we all have. At some point, you should have seen the formation of an eye, and even pigmentation patterns start appearing much later. Does it make sense? So these are things that seem to appear, like for a statistical physics, these are shocking things. How the hell is it possible? I learned everything will become homogenized and uniform. But here is something where from nothing, something is coming up. That's very shocking. We'd like to understand how and why. So one way is to say, there are genes and there are proteins and that does the job. I'm a little finicky guy, so I ask, I'll give you genes and proteins, tell me how to build a fish. I would like the detailed dynamics of that. And can we follow that in space and time? Can we write theories? Can we do experiments and see if that works? So there are these kinds of problems that I'm fascinated by in developmental biology. So there are problems of, how do you make different kinds of cells? You must have heard of things called differentiation. It's the same genome that we all have in every one of our cells, but not all our cells are the same. The head cells and hair cells and neuron cells and so on. So this is a program where different parts of the genome is read at different time points to make decisions, let's make this a fibroblast, let's make that a neuron, and so on and so forth. That's the aspect where, morphogenesis doesn't seem to play much of a role. It's more of a decision-making, information transfer from the DNA to what kind of a cell, what kind of a proteins to expect. The other problem is how to shape up the creature. Shaping up the creature, typically involves 2 stages. One is axis establishment. As you saw in the movie, I'll show you more movies as we go along. Typically, creatures start out by more or less isotropic, maybe one symmetry broken, but more or less isotropic. So your first job is to, define your head and foot and front and back and left and right. That's when you can place things relatively. So when we teach, we put a XYZ axis. How does the creature know what is XYZ? Right? So the first job is to establish coordinate systems. So the creatures actually do this axis establishment. The second job is to do segmentation. So you have a head and trunk and tail and so on. So different regions get partitioned differently. So therefore, they have different functional uses later on. These are problems where pattern formation comes up very naturally. You see there is a spatial segregation of cells. There is a spatial expression of certain chemical patterns. And the information buried in those patterns is used by creatures to build themselves. So we've all gone through this. There were patterns like this even within us when we were in our mother's womb. And so such patterns have been used to sculpt us. Classic examples where such things have been studied very extensively, again, fruit fly, Drosophila fruit fly. So these are two images of the same movie running at the same time. Each dot here is a cell. So there's a couple of hundred, this is 50 micron scale bar. And as you go through in time, you start seeing, starting from something which was, homogeneous distribution of cells to something where you start seeing the appearance of stripe-like patterns. Can you see that? It turns out these very early stripe-like patterns that are laid out in the fly set out the body plans even for the adult. There's a 1:1 mapping between these kinds of stripes that you see here, the color coding, to the final organs that come up in the adult. So very early pattern formation has influence on how you structure body plans. Axis establishment in many creatures, this is the same movie that I showed you earlier. This is a problem where, two things are being accomplished. One is there is asymmetric cell division. Turns out there's also differentiation. But the asymmetric cell division is driven by certain proteins on the surface of the cell. I'll come back to this problem tomorrow. And these proteins, can you see, they form inhomogeneous pattern of red and blue domains. Start out by typically being more or less red. A blue domain develops. Eventually, there is a blue and red interface. It turns out the blue and red interface is exactly where the cell divides. So that sort of decides which one is the bigger daughter, which one is the smaller daughter. So the polarity pattern also established the head-tail axis. So this is the C. elegans worm, soil living creature. The very first cell division also established which side becomes the head of the worm, which side becomes the tail. Now, such patterns are out of equilibrium by definition. To understand them, lots of things have been done. One field which is promising, which is where many of us work, is what's called active matter, more towards the end of this talk and maybe tomorrow about that. It started out by thinking about collections of motile and elongated objects, which are driven out of equilibrium. So there's local consumption and dissipation of energy, but then there's more broader ways to look at that, particularly in the context of development. Okay, so it started out by thinking about, if there's a bunch of birds, a flock of birds, most of you must have seen videos like this. Must have wondered, is it like a droplet of a fluid? Can I think about it like, a droplet of fluid which is deforming in shape and moving? Such ideas motivated people, starting with Vijaykumar model, discrete thing. Tono too wrote the continuous hydrodynamic description, where you can write down descriptions for the density field, the polarity field, which also acts like a magnetization field, and predict the emergence of such order. I do not expect you to understand these equations. We'll come back to them later on at some point. I just want to show you it's not just words. One can write equations and actually calculate things. All right. So this is the end of the introduction there. So I've shown you patterns. There are plenty of patterns. So there can be patterns in what? You can have space and there's patterns in space and time, and they typically break spatiotemporal symmetry. But patterns of what? There can be patterns in scalar fields like density field, concentration fields, or vector fields like flow, alignment, polarity, or even tensor fields like stress and shape and orientation and so on. Now, so that's an example. So there's density fields, orientation degrees, flows, and polarity fields. Typically in biology, this is coupled with chemical reactions. So the food that you eat via metabolic processes drives chemical reaction networks out of equilibrium. You do not maintain detailed balance. These things get coupled to macroscopic order parameters like densities, orientations, flows, and so on. The food that you eat drives non-equilibrium fluxes here. Because of the coupling, there's large-scale spatiotemporal patterns that come up. All right, so there are plenty of patterns in what and of what. So what? Does biology use them? So I'm just fascinated by this video. I'll show you, but I will never go into touch into this. This is too complicated for me. How many of you have seen this before? Okay, nice. Before I play the video, this is a game. There's apparently an octopus sitting here. Take a look where it, take a guess where it is. Octopuses, cuttlefish, these are masters of camouflage, and they use patterns to camouflage themselves. So there is an octopus sitting here, and you're supposed to guess and tell me, where do you think the octopus is? I'm not going to cheat. It's not out of the frame, and certainly it'll come up. That's not going to happen. It's also not buried underneath the sand and it will suddenly come up. That's also not true, but there is an octopus here. Anybody wants to take a guess? Let's see who is smarter, you or the octopus. Take a guess, take a guess. Bottom left here, okay? Anybody else? It's not buried underneath the sand. It's at the top somewhere. Okay, anybody else? Somewhere there? Bottom, top left? Just below here, this one? Okay. There, okay, fine. This one? This is the one? If you've seen the video, it's cheating. You should not answer it. Where bottom? Here. Bottom of the plant, here. Oh, you're saying it's hiding inside the plant. No, no cheating. It's not hidden anywhere right now. All right, okay. So this is a video that Roger Hanlon made from Marine Biology Labs. One day he was apparently swimming under ocean and collecting videos. And turns out that he had a shocking moment. This is a fantastic video, made about 15, 20 years ago. So we play the video and he goes looking for something. Maybe it's behind the bush, maybe it's underneath the sand. And at some point of time, you go near the fish and, oh goodness, the bush is the octopus. Okay. So the so what question is, well, they use it. Can I what? Yeah, of course. So in fact, I'll sure, definitely I'll play it again. There's in fact, the movie plays in reverse just to see how it develops the pattern. It's still an octopus, still an octopus. You see the scales are coming up, the texture is coming up, the color is coming up. I can still make out an octopus. I just go behind and still the eye is there. There you go. You know what the shocking thing is? Amount of time taken to do this transformation. 2 seconds. What do you think? It's diffusion? How big is an octopus? Few centimeters, few tens. Okay. On scales of few centimeters, can diffusion accomplish these kinds of patterns? Take a call. Yes, no? I don't know. There is some speculation they are controlled by neurons and that's what gives them that structure. You want me to play it again? Seen it back? Oh, yeah. So if you're fascinated by this, there is a previous video called Masters of Camouflage available on YouTube. The cuttlefish is even clever, even clever. It uses even time-dependent patterns to mesmerize its prey and capture it. Okay, so that's amazing. Okay, so with that, I want to go and talk about how to think about patterns. I'll start by stability analysis, reaction diffusion systems. I'll show you Turing patterns. Let's calculate something. No octopuses, simple reaction diffusion systems. Nobody knows how to do octopuses. And then I'll come back and tomorrow I'll talk about more mechanochemical patterns. I'll start with ODEs and then quickly go on to PD systems. So say I have a discrete n degree of freedom system. So I label the dynamical degrees by phi I. And I write down equations for the phi I, where the equations can depend on all the variables. And I goes from 1 to n. So if I define the vector phi, I can write this more compactly, where the vector f is also all the f's combined. One of the notions that's important is the idea of fixed points. So the idea is that there is some phi star such that the time derivative vanishes at that phi star. So from the equation of motion, this literally means that phi star is a solution to this system. Is this something familiar to everybody? So you solve for phi star to get the fixed points. So if I give you an example, d phi dt is sine phi. What are the fixed points? N pi. where n is an integer. Does that make sense? So there can be many fixed points depending on the system of equations that you're looking at. So one thing is to talk about fixed points. The next step is to ask for the stability of the fixed point. What do we mean by stability? You say the following. Look, I'm at the fixed point phi star. I'm going to perturb it a little bit. Then ask, how does the perturbation behave as a function of space? Right? So this is a fixed point. And this is the perturbation. So stability of the fixed point, you assess it by asking how the perturbation delta phi evolves in time. So clearly, if the perturbation delta phi goes to 0 at long times, well, phi star is a stable fixed point. Does that make sense? I can take a fixed point, that is the equation, that is the phi star obtained by d phi star by dt. I'm going to perturb it a little bit and ask if the ensuing dynamics is such that the perturbation decays to 0, so which means the system comes back to the same fixed point, phi star. So this is only asymptotic nature. I'm not looking at the transient part. So it's, sorry. Only the asymptotic stability. So one way to get the nature of the fixed points is what's called linear stability analysis. Here you assume the perturbation is much smaller compared to the fixed point in some sense. If not, put some epsilon somewhere and make it appropriately. So you'd like to ask the stability of the fixed point. So you can do a Taylor expansion of f. You have phi star plus delta phi. So that is f of phi star plus the gradient of f evaluated at phi star dot delta plus r order. So clearly this goes to 0 because phi star is a fixed point. So therefore the leading order term is just something which is linear. So if I plug this back in, I get an equation for the perturbation, which is some matrix L dot delta. With a linear stability matrix, L is such that the ijth element is essentially the derivative of fi with respect to phi j at phi star. Is that clear? Now formally, I can solve this equation and say that the perturbation delta phi at time t will essentially be e times t to the L dotted with delta phi at time t equal to 0. Is that fine? It's a formal solution of this differential. So you'd like to ask around phi star, the fixed point, does the perturbation grow or decay as t tends to infinity? What is that decided by? What is that decided by? Have people seen this or no? Sine of what? Sine of? No, L is a matrix. The eigenvalues of L. So this is actually decided by the eigenvalues of. And if all eigenvalues lambda I are negative, then phi star is stable. The real part of the eigenvalues. Thank you. Does it make sense? If at least one of them has a positive real part, well, there is clearly one unstable direction. If the real parts all vanish, then it's marginally stable, and this level of analysis can't tell you. You have to do a higher order calculation. Two lectures. I want to show morphogenesis. If you give me a whole course, I'll do it. I'll do some Galerkin tomorrow. So one thing I would like, is this visible? No. Say it again. Okay, I'll use white. All right, I'll let come. So one thing that I would like to understand is the stability of fixed points is controlled by the parameters in the differential equation. Now, surely when I wrote down a system of differential equations like that, there's not just the independent variables, but there'll also be some set of parameters. Does that make sense? The existence, the value, and the stability of the fixed points will be governed by the set of parameters. P will be polarity tomorrow, so sorry, I can't do anything. Today P is parameters. So here's an example. So if I say d phi dt is A times phi minus phi cubed. Can you please tell me what are the fixed points? 0 and plus minus root A. What about their stability? So you find out df by d phi. So that is from there, A minus 3 phi square. You evaluate df by d phi at each of the fixed points. So around the 0 fixed point, what is df by d phi? A. And df by d phi around the other two fixed points. So just put a phi equal to plus minus root A here, simplify. Minus 2A. Is that right? Now, here's clearly the following statements that you can think about. For A negative, well, the phi star equal to 0 fixed point exists, but the phi star equal to plus minus root A doesn't exist on the real line. So if I'm looking for real fixed points, There's only one fixed point, and that's phi star equal to 0. Is that stable? Why? Because the Jacobian, the linear stability matrix is also the Jacobian matrix. That's equal to A, and A is negative. So there's only one eigenvalue, and that's negative. So therefore, it is stable. What about A positive? Does phi star equal to 0 exist, first of all? Not the stability, just the existence. It exists? Exists, fine. So phi star equal to 0 exists. Is it stable or unstable? It's unstable. The other fixed points, phi star equal to plus minus root A, do they exist for A positive on the real line? And now are they stable? You see the parameter A controls both the existence, the value, and the stability of the fixed point. So we try to encapsulate most of this in what's called a bifurcation diagram. We would typically plot phi star and some control parameter A. Now clearly the 0 fixed point exists throughout the value of A and is in fact stable for A negative. It exists on the positive A axis, but it is unstable. Does that make sense? So the legend is that this is a stable fixed point. The dashed line is an unstable fixed. What about the other two fixed points, plus minus root A? Do they exist on the A negative side? No. But they do appear when A crosses 0 and they take on root A. And they are stable. Does it make sense? I'm assuming most people have seen this before. This is a classic pitchfork bifurcation, forward pitchfork bifurcation. It can exist in the complex thing, sure. I'm only looking at the real plane. You can draw a phase diagram in quotes. Here, there's only one control parameter, so therefore I only have A. And so when A is positive, negative is all that controls. So on the A negative side, I have the fixed point phi star equal to 0, and there's only one fixed point, so there is monostability. On the other side, when A crosses 0, you have two fixed points, plus minus root A, and there's in fact bistability there. You see that? There are two fixed points, both are stable. I'll leave it to you as an exercise to work out this. So if I change the equation to d phi dt is A times phi minus phi cube plus some h. Please work out fixed points, stability, phase diagram, and so on. Assuming most of you have seen this in the context of buckling. Yes, no? You must have seen. Please work out the phase diagram. And in fact, show that there is a nice phase diagram in the A H parameter space. There are two control parameters here. And there is a cusp bifurcation at A equal to 0. And on this side, there is bistability. And on the other side, there's monostability. One final example. If you have a 2x2 system, if I have a two-component system, there's a qualitatively new kind of bifurcation that can occur. So if I have two variables, phi1, phi2, they satisfy the following. Leave it as an exercise. Do a transformation where you define phi1 to be some rho cosine theta, phi2 to be rho sine theta. Work out the equations for rho and see for yourself that this turns out to be the standard pitchfork bifurcation that we got. And the theta satisfies so this clearly implies that theta, no, theta, not phi. Theta is some theta naught plus omega t. And just like the pitchfork bifurcation case, when A is less than 0, rho, there's only one fixed point. But when A is positive, remember there are two fixed points, plus minus square root of A. You can neglect the minus square root of A because I'm thinking of rho like a radius parameter. There's only one fixed point. If you do the inverse transformation, you should be able to show that phi1 is basically rho, which is root A, cosine omega t plus theta naught, and phi2 is root A sine omega t plus theta. So there's something qualitatively new here. So these were fixed points where the dynamical variables did not change in time. So you got 0 or plus minus root A, but there they just stayed there. So this is a system of equations where the fixed point is actually such that in the rho theta variables, it is indeed time independent. But in the phi1, phi2 variables, this is something which is oscillatory in time. Does that make sense? So if you look at the phase portrait, phi1, phi2, when A is negative, you'll have only one fixed point, which is rho equal to 0. So therefore, both phi1 and phi2 equal to 0. But when A is positive, you'll have what is called a limit cycle. Is that fine? If you give me some feedback, it'll be nice. If it's trivial, I can just skip it. If it's new, I can go slow. If you don't say anything, I have no idea. Yeah. How do you?
The fact that the fixed points are time dependent, without going to rho theta, can you see it? Because there's no.
You try to solve these equations. See if you can find time independent solution. These are cubics, not so easy to solve, algebraic mess. Okay. At the end of the day, you'll find some transformation where things will simplify, and this is the simplest.
I see. Okay.
For people who know what I'm writing are what are called normal forms, the full equations might be something very complicated, but near the bifurcation, you can show that you can write them in these standard forms. So this is what is called a Hopf bifurcation. So this is a bifurcation that happens when a pair of eigenvalues of the linear stability matrix cross the imaginary axis. Does it make sense? If you work this out, you'll find that the real part is positive, that is A positive, but there is a non-zero imaginary part. That's what gives you the opposite. So in general, for a system of two equations, with 2 degrees of freedom, I can always write the equation for the perturbations as the linear stability matrix L into delta phi. I want you to remember this because I'll need this. If I have a 2 by 2 matrix L, can you tell me how the eigenvalues are related to its trace and determinant? I'll need this for the Turing in a few minutes. So I define T to be the trace of L and delta to be the determinant of L. How are the two eigenvalues related? Trace plus minus square root of trace square minus 4 times determinant. Okay. Is this fine? I want to do a similar analysis for PDE systems, partial differential equation. Remember, pattern formation essentially involves PDEs. I want to go there and develop that. While I erase, if you have questions, you can bug. There are other ways to find the stability of systems, which are more complicated. One of the things that Suraj mentioned about modal analysis. There's another complication that if the linear stability matrix is not self-adjoint, you have to be careful. Just its eigenvalues may not tell you the stability. There can be algebraic growth at short time scales. So I'm not looking at that. I'm only asking for the asymptotic stability, that is stability at long times, just given by the linear stability. Okay. I assume most people have seen random walks and the connection of random walks to the diffusion equation. Yes? Yes? No? Okay, great. So I want to look at stability analysis for partial differential equation systems. So let's start with diffusion. So typically, you think about a coarse-grained description of a bunch of particles moving around. You measure the concentration field, number density, whatever you want, which is a function of both space and time. Let's say here the concentration. And by whatever way, either appealing to Fick's law or starting from random walk and doing some coarse graining, you can show that the concentration field satisfies what's called the diffusion equation. Is everyone familiar with this? Okay. How do I do the stability analysis of such system? I will also add a simple linear degradation reaction. So in the absence of diffusion, the concentration will go to the value C naught, and the rate is set by the turnover rate K. So surely C equal to C naught is a fixed point. Is that right? If I put C equal to C naught, the diffusion term vanishes and the reaction term also vanishes. In fact, this is a homogeneous fixed point. That means there is no spatial dependence. So remember, C is a field here, which depends both on space and time. And so therefore, it is possible that one could have inhomogeneous fixed points in general for some partial differential equations. Not for this, but the ones that I'll discuss later. The first thing you notice is that C naught is a fixed point. In fact, a homogeneous fixed point. What about its stability? How do we assess the stability? We do the same trick as we did earlier. We look at the fixed point, we put in a perturbation and ask how does the perturbation go to 0. But then what perturbation to put? Remember, C is a field here. So if I have the concentration field as a function of x, so I have some C equal to C naught value. So that's my fixed point. For now, I'm not specifying the extent of the domain or the boundary conditions. I'll come to that in a few minutes. For now, assume it's a finite size system here. So I have a uniform fixed point. I have a homogeneous fixed point. I want to assess its stability. How does one do that? Perturb it. Perturb it where? Perturb it everywhere. So maybe raise the value everywhere, lower the value everywhere. Is that the only kind of perturbation? Huh? Louder, I can't hear you. Plane wave perturbation. So maybe I can put a perturbation to find some different color. I can put a spatially dependent perturbation. You can cook up any spatial kind of perturbation that you can. So how do I actually try to analyze for every possible spatial perturbation that I do? How do I analyze the stability of it? How would one do that? Any ideas? If you have seen this, let the students figure it out. People have seen this. I can do very fast. I will disregard the first row. It doesn't count. It's not fair. People have seen this, not seen this? Okay. Okay. So then let's do it. Surely you have to put in a perturbation that depends on space. Does that make sense? Now, what kind of perturbation to put? One way is that I do an expansion of C in the spectrum of some operator. Physically, the way to think about it is that you give me any arbitrary perturbation, I can represent it as a Fourier series or a Fourier transform if it's an unbounded domain. And so I would like to assess the stability for each mode, each Fourier mode. And then the stability statement will become that for all modes, if the perturbations decay, then the system is stable. If for any mode the perturbation grows, clearly the system is unstable. If you remember, when we had an n degree of freedom, we demanded that the stability means that all the eigenvalues should be negative, real part should be negative. Even if one of them has a positive real part, well, that's an unstable direction. So there you will grow, but because the system is coupled, well, surely it will make all the other things as well grow in general. So the way you would analyze this is by asking, can I do this by doing a perturbation in some spectral space? So clearly I can do that, right? So I can do delta C as a function of space and time. I'll use the same symbol distinguished by the argument. I'll do a Fourier transform of this kind. I'll plug that in, and what happens to derivatives and the Fourier transforms? Q square, right? So can you show that the equation for the qth Fourier mode, which will turn out to be just omega q, well, just let me write that out. This will be just dq square delta cq minus k times delta cq. Is that obvious? Everyone? What does this imply? Immediately, that delta C Q at time t is essentially e to the minus dq square minus k, the whole thing into t into delta c q. Okay, I should have done subscript. Anyway. So what happens to the perturbations? Is there any mode where delta CQ grows with time? Remember, the diffusion constant is positive, the turnover rate is positive. So in other words, the frequency omega is essentially dq square. Immediately notice that omega q is in fact real and is negative for all q. Implies the system is stable for all perturbations. Is that fine? Oh, yeah. Thanks. Thank you. IQX. Maybe a plus, maybe a minus. Plus. Is this fine? So remember, the system was already linear. I mean, linear diffusion, linear turnover. So there was no need for a linear analysis, linear stability analysis. All we did was a Fourier transform. And so you find out that every mode is such that the growth rate is negative for all modes. So therefore, the system is stable, which is the intuitive picture, right? No matter what perturbation I do, diffusion will try to homogenize things. So the other exponentially decaying reaction is something which will just give you a mode independent relaxation rate, and so therefore everything will homogenize at long time. Standard intuitive picture that we all have, diffusion homogenizes stuff. Is that fine? I'll leave it to you as an exercise. Do a similar thing for the example that we saw earlier, but this time including spatial degrees of freedom. So I have some field phi, which depends on space and time. And I tell you the equation for phi is diffusion minus A times phi, or plus A times phi, minus, this is a nonlinear equation. The non-spatial part we've just analyzed. So I've just added diffusion to this. So you go back and do the same analysis that you did. First, you find the homogeneous fixed points. And around each of the homogeneous fixed points, you put in a perturbation that depends on space and time, expanded perturbation in a spectral fashion, plug that in, assume the perturbation is small, so you can approximate the phi cube term, and then do the calculation like what we did earlier, and find out for yourself which of the fixed points are stable, which of the ones are unstable, and does diffusion do anything non-trivial. Can I go ahead? We stop at 12:45? I'll be able to. Yeah.
What happens if we have a point source there in C0?
Just hold the thought for like half an hour, I'll show you that.
Okay.
You will work on that's a relevant biological example. Anything else? Okay. So I want to show you something slightly non-trivial, what's called the Turing instability. This is not good. This is the same Alan Turing. The paper 1952, chemical basis of morphogenesis. So here's the fundamental question that he asked, and the reason why his ideas were not taken seriously for a long time. He asked the following. Say, we have a system that is stable in the absence of diffusion. He asked, can diffusion destabilize the homogeneous state? Is the question clear? Usually you think about diffusion as something that has a stabilizing effect. Like the example that we saw, no matter what perturbation you do, if the system was stable in the absence of diffusion, here without diffusion, it's something where it uniformly, it goes to C naught at the rate K. Addition of diffusion didn't do anything. Maybe it modified the growth rate, but then everything was eventually stable and the whole system became uniform at long times. I'd like to ask if there is a possibility that you can have a system which is stable in the absence of diffusion, and adding diffusion makes it unstable. Is it possible intuitively? That adding diffusion actually makes the homogeneous state unstable. What do you think intuitively? Or single field is not possible, but even with multiple fields? Say it again. You need cross terms. Of course, you will have a coupled system on that. But is it intuitively obvious, or does it make sense that adding diffusion can actually destabilize something? And the intuitive picture is, at the basis of diffusion is random walk. If there's unbiased random walk, everything should homogenize at long times. So you're asking a question that, look, I don't have diffusion, so there are no spatial degrees of freedom. So therefore, there is a system which has some steady state, and that steady state is stable without diffusion. I'm just going to add diffusion. You can move around in space. Claim is, the answer is yes. The claim is that this system can actually become unstable due to diffusion. So it's a diffusion-driven instability. Weird, no? No wonder he didn't take him seriously for a long time. So answer is yes, and I want to work that out in the remaining part of the. So can I go ahead? Is this clear? Or is it your tummy is telling you it's hungry? And forget the science. So one class of patterns that, one class of systems that show very non-trivial patterns are what are called reaction diffusion patterns. And Turing systems typically fall in that category. So when I go to the presentation, I'll show you some more examples. So by definition, these are systems where there is a diffusion term and some reactions. So the reactions change the numbers of the individual components, The diffusion just tries to move them around whenever there are variations in the concentrations. So this is very general, but I'll take something very simple and show you something non-trivial can happen. So Turing said, let's do the following, or I'll paraphrase Turing and do it in a slightly different language. So let's say you have a two-component system with the following equations. Is that clear? So phi1 and phi2 are two concentration fields, any two variables. They diffuse with diffusivities d1 and d2, and there's some reactions f1 and f2, which depends on both the variables, phi1 and phi2. So say we found a fixed point, a homogeneous fixed point. A stable, homogeneous fixed point. Phi star, which is phi1 star, phi2 star. And as usual, I want to do a perturbation and find out if the perturbation can actually grow. As before, we'll do a Fourier transformation. A bunch of implicit assumptions here. I'm just saying Fourier transform, it's fine. The point is that you must have a homogeneous fixed point. So if you have an inhomogeneous fixed point, you cannot use plane wave modes as the basis. That's not possible. 2 lectures, I can't do all those things. So you define the Fourier transform of the perturbation in position and time, just like what we did earlier. Yeah, why am I writing it? I've written there. It's exactly the same. And you do a linear stability analysis. So I claim that the amplitudes phi1 and phi2 will satisfy the following equation. Where the linear stability matrix L is A11, B1 Q square, A12, A21, A22 minus B2 Q square, where Aij is defined to be the derivative of Fi with respect to phi j evaluated at phi equal to phi. See if you can see through this, please. If you do not have the diffusion terms, just like we did in the first part of the lecture, you would basically have the linear stability given by a Jacobian matrix, where you take the Jacobian of the reaction terms at the fixed point phi star. So that's nothing but the derivatives of the Fi with respect to the phi j evaluated at phi star. So that will be the terms which are independent of Q. But you just add the diffusion, and just like that example there, all you will get is a term that involves the diffusion constant and the wave number square. Does that make sense? Can people see through this? So we started out by asking that I want the system to be stable in the homogeneous case, in the absence of diffusion. So when diffusion is absent, or when you're looking at the homogeneous case, this is basically the zero mode. Does that make sense? Q equal to 0 is the homogeneous mode. So what do I know about this? if I define the trace of the linear stability matrix, call this LQ, and the determinant by TQ and delta Q, this condition implies that I want the trace at Q equal to 0 to be negative and the determinant to be positive. Can you please look at that and I erase that? You have the two eigenvalues of the 2 by 2 system in terms of the trace and the determinant. Just see that if these conditions are satisfied, you're guaranteed that the real part of both the eigenvalues will be negative. Yeah. Sure. Thanks. Is this obvious? If the trace is negative and the determinant is positive, both the eigenvalues have negative real parts. It's just some quadratic equation picture, nothing else. Is that true?
Said that it's stable in the absence of diffusion. So shouldn't we look at D1, D2 being 0? I don't understand why we're looking at Q being 0.
Equivalent when Q is 0. No? So all that survives is the reaction part of the system, which is just the A1, A2s. So can you stare at the matrix and tell me that is when Q is 0, what do these two conditions imply? A11 plus A22 should be less than 0. And one more condition. A11 into 22 minus 12 into 21 should be greater than 0. I will choose A11 to be positive. It's up to you. You can choose A22 to be positive. It doesn't matter. It just swaps the role of phi1 and phi2. Immediately, what can you tell me? A22 should be negative. One more condition. The product of the cross terms, 1, 2 into 2, 1. Negative, positive. So all this is just asking that the homogeneous state be stable. That is, if a system has a stable steady state under the influence of only the reaction terms, no diffusion. These are the conditions. Does that make sense? This is just like a standard 2 by 2 ODE system that I talked to you about. Yeah. I mean, if you choose both negative, then there's nothing. I can choose A22 to be positive as well. Doesn't matter. Why did I break 1 and 2? No reason. Choose A22 to be, you'll get something similar. Absolutely no reason. Both negative won't do the job. I mean, you won't get anything eventually. Do the following. I mean, choose all possible combinations and see if you choose both negative, I mean, everything will be stable. Can I go ahead? I want to ask if I can get an instability or a Q not equal to 0. What is the trace for a wave number Q from the full matrix, not at Q equal to 0? Stare at it and tell me. 1, 1, 22 minus do you see that this is already less than T0? This is T0. As far as I know, diffusion constants are positive. We'll be in trouble if they're negative. Q is Q squared, so therefore this whole thing is positive, but with a minus sign. So this is already less than T0, which you remember is already less than 0, because I want the homogeneous state to be stable. So if I want an instability, I want the following, that there must be some wave number Q such that the real part of omega Q is positive. Now, PQ is already such that it's less than T0, which is less than 0. So this is possible only if you violate the other condition, that is the determinant being positive. So if you violate that and make the determinant non-positive, then you might get an instability for a non-zero Q mode. Does it make sense? Now, what is delta Q? So it's A11 minus D1 Q square, A22 D2 Q square minus A12 A21. I want this to be less than or equal to 0. Is it visible if I write here? Yes, no? So the threshold condition, that is, you find out when delta Q is just 0. You can easily find that out if you look at the structure of delta Q. If you do the expansion, there'll be some things with Q square and there'll be something with Q power 4. The Q power 4 will come with a positive sign. You would like to find out the optima of delta Q. And that's easy to do because all I can do is take delta Q, differentiate that with respect to Q square. That will find the optima. Set that equal to 0. That tells me what is the Q at which that's a optimum. If you do that, you find D1, D2 Q square minus D1 A22 minus D2 A11 will be 0. Shouldn't there be a 2 where? I'm differentiating with respect to Q square. So where should be a 2? 2 D1. You're right. Fine. So solve from here, you find that Q0, I'll call this Q0. There should be some Q0 for which this works out. So that Q0 square should be half A11 by D1 plus A22 . Remember, Q0 square is a wave number square, so that's definitely a positive number unless Q0 is 0. We've also taken A11 to be positive, and that has implied that A22 should be negative. All that was just from the stability of the homogeneous state. So if I define two length scales, L1 and L2, L1 square is D1 by A11. L2 square is D2 by modulus of A22. And you see that Q naught square is half 1 by L1 square minus 1 by L2 square. From here. Make sense? So the answer to the question that we asked, if diffusion can destabilize the homogeneous state, the answer is yes. If A11 is positive, A22 is negative, and the Q naught square that I got there should be positive, which means L1 should be less than L2. If you resubstitute the coefficients, this means D2 by D1 should be greater than A22 modulus divided by. Okay, so with the algebra done, let's get some physics intuition.
So here we have only shown that this is the optimal Q, but we haven't shown that it is unstable for this Q.
Yeah, yeah.
Right?
So you ask that this is the optimum, plug it back into the, where is that determinant somewhere? Here. And you would want the determinant to vanish at that point.
Yes.
That gives you this condition if you want. Take this.
Yes.
With L1 greater than L2, plug it back, and you can show that delta Q will be greater than 0. The optimal condition is this. I just looked at the peak value. Okay. Plug that in, and that will show you delta Q is 0. Okay. What do those two length scales L1 and L2 mean? So right from here, can you see that there is a length scale which is square root of D by K? Yeah. So this is a length scale on which, if you have a perturbation in C, that information is carried through. It's like a correlation length that you want to think about. So this is for a one component system. If you notice the definitions of L1 and L2, they're exactly the same. A11, A22 are the derivatives of the F1 and F2 with respect to phi1 and phi2. So they're basically the analogs of the K that I had there. So the L1 square, L1 and L2 are exactly the analogs of the L that I defined for the two. So it turns out all you need is this should be negative, which you already know, because all you demanded was the stability of the homogeneous state. Where are you talking second term? Delta Q, yeah? No, hold on. A22 is negative. Work out for the case just equal to 0. That's enough. Stability is when it's positive. The threshold condition is when it's just 0. Yes? Yes? Or negative. This is already negative. Without the minus sign, it's already negative. Yeah. Can you just plug this in there and do the algebra? If it doesn't work out, I can do the algebra on the board. I just want to end with the physics. So the condition that you got here says the following. That if you have an activator, so the species phi1 is such that A11 is positive, so which means it makes more of itself. Does that make sense? Phi1 increases phi1 more. D phi by dt, some positive things times phi1. And Phi A22 is negative, you have an inhibitor. You will get an instability when you have short range activation and long range inhibition. So the range is essentially the value of the length scales L1 and L2. So you want the activator length scale to be small compared to the length scale of the inhibitor. Does it make sense? Intuitively, it may not make sense, but just see the algebra first. The algebra works out, then we'll figure out by the physics. Turing showed this, and it took a hell lot of a time for people to trust because there were no experimental proofs. People constructed synthetic chemical systems sometime in the mid-90s and showed that this instability does exist. It's only in the late 90s and early 2000s people found evidence of this in real biological systems. I'll show you in the next lecture that fish stripe patterns were thought to be Turing patterns, something that was speculated for a very long time but without proof. Sometime in 2014-15, people actually showed mammalian digitation patterns are Turing patterns. Your fingers are actually a result of a Turing pattern, at least in mice. I assume in us too. All right, I will stop there for today. Tomorrow I'll pick up from there and start. Thanks.